This lecture will cover Chapter 4, Cellular Activities and Clinical Disorders of Innate and Adaptive Immunity. And here are some basic terms that you should be familiar with just in order to better understand this lecture. Neutrophils as well as monocyte macrophages are the principal cells that will defend the body against various types of microbial invasion, as well as produce a localized inflammatory response. The granules inside of the neutrophils contain various antimicrobial substances, so during the phagocytic process, these enzymes are released and they will actually disrupt the integrity of the cell itself, ultimately destroying it. Phagocytosis is divided into 6 stages. You have chemotaxis, adhesion, engulfment, phagosome formation, fusion, and digestion and destruction. The physical occurrence of damage to the tissue, whether it's by trauma or some sort of microbial multiplication, actually releases substance such as activated complement components and products of infection that will actually initiate the phagocytosis. In addition to phagocytosis, which also includes the release of antimicrobial molecules at the site of infection, there has been another defense mechanism that has also been discovered. So this mechanism is the formation of neutrophil extracellular traps, otherwise known as nets. These are actually produced after the release of the nuclear contents of the neutrophil into the extracellular spaces. Nets function in innate immunity, and they are composed of chromatin components, including histones and neutrophil antimicrobial proteins. Microbes can then be trapped inside of these nets where they will encounter the high concentrations of antimicrobial proteins. Before we proceed, let's first put a visual image to the process of phagocytosis. So various phagocytic cells will continually circulate throughout the blood, the lymph, the GI tract, and throughout the respiratory tract. So when there is some sort of trauma that takes place, the neutrophils will arrive at the site of injury and can be found in the initial exudates in less than 1 hour after that trauma has actually taken place. So monocytes are slower to actually move into the inflammatory site, but macrophages are already in place within the tissue of the body, so they are able to deal with any sort of intruding agent a little bit faster. It has been shown that neutrophils at the site will actually release myeloperoxidase, which will facilitate recruitment of other cells to this area to actually start this process. Neutrophils have also been shown to activate complement when stimulated by any sort of cytokine or coagulation-derived factors. So neutrophils activate the alternative complement pathway, and then they will actually start to release C5 fragments. And then from there, you will see a further amplification of the neutrophil pro-inflammatory response, and then you will start to see other neutrophils that will quickly start to gather in this area of injury, and later you will start to see the monocytes, macrophage, and the dendritic cells that will also come into place, where the macrophages and the dendritic cells will help with the cleanup of the injury and the dead host cells. So chemotaxis will actually start to take place once all of these chemotoatic factors or actually start to become present in the area. An example of these would be the chemokines. And so when these are released, there are at least 50 different cytokines that are involved in cell migration, activation, and chemotaxis. And so the coating of the organism by the various molecules will actually speed up the phagocytosis process, and this is called opsonization. And this is where you've had those antibodies that have kind of coated the outside of that particular cell or that microorganism, and then the FC portions of the antibodies and the complement are activated, which actually kind of sends out those signals that, hey, this is a cell that needs to be destroyed. Then these targeted cells will start to adhere to the various phagocytic types of cells, and various other chemicals will start to be released, and then you will start to see the inflammatory response that will begin, and those various inflammatory chemicals will start to be released in the extracellular fluid. So some of the sources of these inflammatory mediators include things like histamine, prostaglandins, cytokines, that were all released at that particular site of injury. And of course, this will all promote inflammation. From there, the phagocyte will continue to engulf the cell and pull it inside of the cell, forming a pseudopod, which will actually kind of form around the pathogen where it's pulled inside of the cell because of that interaction between the Fc receptors and the Fc antibody portions on that particular bacteria that has been coated, or that particular cell that's been coated. So once it is inside the cell, that vacuole is what we refer to as a phagosome. Now once it is inside the cell, the process of digestion and destruction begins. And you have all of these various granules inside those cells that will start to actually break down that particular targeted cell. There are several neutrophilic disorders that can happen. We're not going to go over these in a lot of detail because they were covered in hematology, but we are just going to kind of add to that a little bit from the immunology standpoint. So although neutrophils can provide the major means of defense against bacterial and fungal infections, they can also be destructive to host cells. So those same oxidative and non-oxidative processes that actually destroy microorganisms can affect adjacent host tissue. In addition, there are also many autoimmune diseases that are thought to be caused by the inappropriate activation of the process of phagocytosis or ineffective resolution of the inflammatory process. And so this is when the body will actually attack its own cells and tissue. Patients that have quantitative or qualitative defects of the neutrophils tend to have a higher rate of infection, which illustrates the importance of the neutrophil to the actual body's defense. So individuals with a marked decrease of neutrophils, otherwise known as neutropenia, or that have severe defects in the neutrophil function, will frequently have recurrent systemic bacterial infections, as well as disseminated cutaneous pyogenic lesions and other types of life-threatening bacterial and fungal infections. Occasionally, you can see a small number of patients that have congenital abnormalities of the neutrophil structure and function. And so those are all listed here. I'm not going to go over them in detail because we did cover those in hematology. Functionally, monocytes and macrophages have phagocytosis as their major role, but these cells perform at least 3 distinct but interrelated functions in a host defense. So the categories of host defense functions of monocyte macrophages include phagocytosis, antigen presentation and induction of the immune response, as well as secretion of biologically active molecules. So the principal functions of the mononuclear phagocytes in the body's defense result from the changes that actually take place in these functions when the macrophage is actually activated. So macrophages carry out the fundamental function of ingesting and killing any sort of invading microorganisms, such as intracellular parasites like the Mycobacterium tuberculosis, as well as some of the fungi. In addition, macrophages remove and eliminate eliminate extracellular pathogens such as pneumococci from circulating within the blood. They also have the ability to phagocytize particulates and aggregated soluble materials. So this process is actually enhanced by the presence of receptors on the surface of the Fc portion of the IgG and complement C3. So the ability to internalize soluble substance supports the increased microbial and tumoricidal ability of the activated macrophage. Now, another important function of the macrophages is their ability to actually dispose of damaged or dying cells. So macrophages will actually line the sinusoids of the spleen and are particularly important in ingesting the aging erythrocytes. They have also been involved in removing tissue debris, repairing wounds, and removing debris as embryonic tissues replace one another. Phagocytic activity actually increases when there's any sort of tissue damage and inflammation, and so this will actually release substances that will attract other macrophages. Now, the activated macrophages will migrate more vigorously in response to any sort of chemotoxic factors, and then they will actually enter the site of inflammation. And so now keep in mind that inflammation can include anything like an infection as well as any sort of cancer type of cells. Monocyte and macrophages have been shown to be abnormal in a variety of diseases. The abnormality is partial and no related association with increased susceptibility to infections has been established with these disorders. In cases of severely depressed migration of monocytes, however, it is likely that the dysfunction predisposes a patient to infection because of other defects of the host's defense system coexist with these diseases. Now here we have a nice chart that kind of depicts what the abnormality is and what the disease association is. Qualitative disorders of monocytes and macrophages manifest as lipid storage diseases include a number of rare autosomal recessive disorders. The expression in macrophages of a systemic enzymatic defect permits the accumulation of cell debris that is normally actually cleared by the macrophages. Macrophages are actually very prone to accumulating any sort of ungraded lipid products. Resistance to infection can be impaired, and at least partially, because of a defect in the macrophage functions. So disorders of this type include Gaucher's disease as well as Niemann-Pick disease. Now I'm not going to go over these in a lot of detail because again this was something that was covered in hematology. Now, if a patient has a leukocyte adhesion deficiency, then ultimately this will lead to recurrent and often fatal bacterial and fungal infections. Acute inflammation is defined as an immune response resulting in complete elimination of a pathogen, and it is followed by resolution of tissue damage with the disappearance of leukocytes from the tissue and then full regeneration of tissue function. Sites of acute inflammation will actually have more neutrophils and activated T lymphocytes than a site of chronic inflammation. With chronic inflammation, the immune system tries to actually contain an infection and then minimize the tissue damage. Sites of chronic inflammation have an abundance of macrophages, cytotoxic T lymphocytes, and even B lymphocytes. As you can see depicted in this image, in acute inflammation, tissue damage results in inflammation, a series of biochemical and cellular changes that will facilitate the phagocytosis of invading microorganisms or damaged cells. Now, if inflammation is sufficiently extensive, it is accompanied by an increase in plasma concentration of acute phase reactants. An inflammatory response involves at least three major stages. So first you have the dilation of the capillaries that will actually increase blood flow to the area. Then you will have microvascular structure changes and escape of plasma proteins from the bloodstream. And then you will have leukocyte transmigration through the endothelium and the accumulation at the site of the injury. Now, if an inflammation overwhelms the entire body, then you have what is called a systemic inflammatory response syndrome. So sepsis or severe sepsis and septic shock are progressively severe stages of the systemic inflammatory response syndrome. Biochemical markers that we associate with sepsis include tissue necrosis factor in your interleukin 1 and interleukin 6, which are pro-inflammatory cytokines. Other proteins that are produced include procalcitonin and chemokines. Additionally, you may see if there's inflammation in the liver, you will see C-reactive protein actually be produced. So again, we've covered these in a previous lecture where we discussed the testing for procalcitonin and C-reactive protein. So again, you can kind of see how the body responds and produces those, which allows us to detect them through traditional laboratory testing to be able to be able to detect when an inflammatory type process is starting and hopefully prevent sepsis. So a cytokine storm was mentioned in a previous lecture, but we're going to go over it a little bit again. So there is something called a cytokine storm syndrome, and these are a group of disorders presenting a variety of inflammatory disorders that will result in an overwhelming systemic inflammation, as well as unstable blood flow in the heart and vessels to profuse normal organ function, multiple organ dysfunction, and potentially even death. Now, something referred to as macrophage activation syndrome is actually a type of cytokine storm syndrome. With macrophage activation syndrome, you have macrophages that are an innate immune cell that will sense and respond to microbial threats by actually producing an inflammatory molecule that will eliminate the pathogen and promote tissue repair. However, a dysregulated macrophage response can be damaging to the actual host cell. So the main pathophysiological feature of the macrophage activation syndrome is the fact that there's excessive activation and expansion of cytotoxic CD8 positive T cells in macrophages. So this activated immune cell produces large amounts of pro-inflammatory cytokines, creating a cytokine storm. So nonspecific acute phase reactants with an abrupt decrease in circulating peripheral blood cells and an acute drop in fibrinogen are actually associated with macrophage activation syndrome, but can also be seen in any cytokine storm syndrome, and it actually often suggests that there's an active DIC that is present. As you may previously recall, the principal cells of the adaptive immune system are the T lymphocytes and B lymphocytes. These categories of lymphocytes can recognize foreign antigens, directly destroy some cells, or produce antibodies as plasma cells. The total immune response involves the interaction of many different cell types and cell-mediated and antibody-mediated responses. So T lymphocyte subsets, including natural killer cells, together with classic innate immune cells, contribute significantly to the development and establishment of acute and chronic inflammatory diseases. The introduction of monoclonal antibody testing has actually led to the present identification of surface membrane markers on lymphocytes and other cells. So these surface markers can actually be used to identify and enumerate various lymphocyte subsets or to establish lymphocyte maturity, as well as classify types of leukemias and lymphomas and monitor patients in an who are actually on immunosuppressive therapy. Cell surface molecules recognized by monoclonal antibodies are called antigens. So because antibodies can be produced against them. Originally, surface markers were named according to their antibody that they actually reacted with, but now we've developed a more common nomenclature system, which is called the cluster differentiation, otherwise known as CD. So the CD numbers have been assigned to more than 320 unique clusters and subsets of monoclonal antibodies. So the CD system can actually identify and discriminate between different cell types or populations, and it can actually differentiate the maturation stages of the lymphocytes. So cell membrane markers can actually be categorized different ways. So some markers are specific for cells of a particular lineage or a maturation pathway, and some markers may vary expression depending on the site of activation or the differentiation of the cell types, such as when we actually do CD antigen identification to classify lymphocyte subsets such as CD4 and CD8. Now, in addition to the use of CD classification for the identification and separation of the lymphocytes, CD antigens are involved in various lymphocyte functions. This includes promotion of cell-to-cell interaction and adhesion, and transduction of signals that will actually lead to lymphocyte activation. So today, the use of monoclonal antibody testing and flow cytometry It's pretty much the gold standard for diagnosis and treatment of various malignancies. So as we learned in hematology, actually being able to immunophenotype the different malignant T and B cells allows us to be able to classify the leukemias and lymphomas. And in this slide here, you can see an image of some of the various CD markers on the lymphocytes. I'm not expecting you to know these, but just be familiar with these markers. Actually kind of differentiate the difference between the T and the B and different stages that they are in. So your textbook does have a great little picture here that kind of depicts the phases of adaptive immune responses. So an adaptive immune response consists of distinct phases. The first three are the recognition of the antigen. Then you have the activation of lymphocytes, and then the elimination of the antigen, which is the effector phase. The response starts to decline as the antigen-stimulated lymphocytes will die by apoptosis, which will start restoring hemostasis, and then the antigen-specific cells that survive are responsible for the actual memory. Now, the duration of each of these phases can vary just dependent on the individual's immune response, but you can see here about a general approximation of what happens when. So these principles apply to humoral immunity, which is measured by your B lymphocytes, and cell-mediated immunity, which is mediated by the T lymphocytes. So to further elaborate on the previous image, so when you have virgin lymphocytes that will encounter an antigen, they will actually proliferate and differentiate into effector T cells. This includes the cytokine-secreting CD4 positive helper T cells and CD8 positive cytotoxic T lymphocytes. Effector B lymphocytes are antibody-secreting plasma cells. Some of those antigen-activated T and B lymphocytes will actually differentiate into memory cells that survive for longer periods. Now, these memory cells are actually responsible for the rapid and enhanced response to any sort of previously encountered antigen. Your memory B cells will carry surface IgG as their antigen receptor, and your memory T cells have an increased level of cell adhesion molecules that function as chemical mediators in any sort of inflammatory process throughout the body. Now, persistent antibody titers that are actually directed against specific antigens are derived primarily from long-lived plasma cells. So primary and secondary immune responses will generate a separate pool of long-lived plasma cells in the spleen, which will actually migrate to the marrow, where they will occupy essential survival niches and then can persist for the life of the host without the need for self-replenishment or even turnover. The marrow plasma cell doesn't actually require any sort of ongoing contributions from the memory B cell pool for its actual maintenance. But when depleted, plasma cells are replenished from the pool of memory B cells. Persisting antigens, cytokines, or toll-like receptor signals may actually drive the memory B cell pool to chronically differentiate into long-lived plasma cells for a long-lived antibody production. Now, most of the lymphocytes that we find circulating in the blood are T cell derived from bone marrow progenitor cells that will actually mature in the thymus gland. These cells are actually responsible for cellular immune responses and are involved in the regulation of antibody reactions in conjunction with the actual B lymphocytes. Differentiation of the lymphocyte begins in the thymus with the actual thymocyte. Now, early surface markers on the thymocyte that are committed to begin T cells will include the CD44 and the CD25. So as the thymocyte develops, there is an orderly rearrangement of the genes coding for an antigen receptor. Now, maturation is a very complicated process, but it lasts for about 3 weeks. Now, during this period, cells will filter through the cortex of the medulla of the thymus. Thymic stromal cells, which include fibroblasts, macrophages, epithelial cells, and dendritic cells, all of these cell types play a role in the actual T cell development. Now, in testing, the early thymocytes that actually lack CD4 and CD8 surface markers are referred to as double negative thymocytes. Cells that are positive for CD4 and CD8 are referred to as double positive. and they represent the second stage of the thymocyte development. Now, some of these cells will actually die within the thymus, but there is a second selection process called a negative selection that will take place among the surviving double positive T cells. Now, this whole process of clonal selection accounts for most of the properties of the adaptive immune system. So mature T cells that have actually survived that selection process will then leave the thymus with only one marker, CD4 positive or CD8 positive. And from there, they will migrate to the medulla and then gain functional maturity in blood circulation. Now, the antigen receptors in these lymphocytes is very large and diverse, with an increased probability of encountering an antigen that will actually bind to its receptor triggers to activate and proliferate that particular cell. Now, T lymphocyte subset divisions are not absolute, with considerable overlap or redundancy in the function among the different subsets. So CD4 T lymphocytes represent a population of cells that are central to the protection against a wide range of pathogens. There are two subsets of activated CD4 T cells that are Th1 and Th2 cells. Now, I'm not expecting you to know anything really about these. Just know that there are some subsets that are even further divided with your lymphocytes. So there are actually some CD4 T cell populations that are actually distinct lineages of cells that have already distinguished from one another when they actually emerge from the thymus. And so these are things like natural regulator T cells and natural killer T cells. There are several subtypes of lymphocytes that represent alternative patterns of differentiation of naive CD4 T cells. And they are essential for the differentiation processes that are essential to the monitoring of effective and regulated immune responses. Your helper T lymphocytes, which we refer to as your T helper cells, play a critical role in orchestrating the adaptive immune responses, and they can also be assigned to three major CD4 effector subsets. Cytotoxic T lymphocytes, or T cytotoxic cells, are effector cells found in the peripheral blood that are capable of directly destroying virally infected target cells. So most of your T cytotoxic cells are CD8 positive and recognize antigens on the target cell surface associated with the major histocompatibility complex class 1 molecules. And so this would be like your HLA types A, B, and C, as well as the major histocompatibility complex class 1 alone. Your suppressor T lymphocytes are functionally defined T cells that have been downregulated as far as their actions of the other T and B cells. So these cells have really no unique markers. So defining the properties of memory CD8 T cells includes being present in higher numbers than those virgin CD8 T cells, and the ability to mount a rapid secondary response with a lower response threshold. Innate lymphoid cells are lymphocyte-like cells that produce cytokines and perform similar functions to CD4 positive and CD8 positive effector cells, but they don't actually express that T cell receptor. Their major function is actually to provide an early defense against any sort of infectious pathogen, to recognize stress and damage of the host cell, and to help eliminate these cells. They also influence the nature of subsequent adaptive immune responses, and the first and best characterized innate lymphoid cell would be the natural killer cell. These cells will actually kill infected and damaged cells. Now, natural killer lymphocytes have an associated effector function similar to T lymphocytes, but they still lack that T lymphocyte antigen receptor. They actually destroy target cells through a non-phagocytic mechanism called a cytotoxic reaction, and they are essential mediators of virus immunity. Clonal expansion happens after there has been a viral exposure. B cells will mediate and regulate many other functions that are essential for immune hemostasis, including an antibody-independent pathogenic role of the B cells, which gives them the capability to present an antigen. They regulate wound healing and transplanted tissue rejection, and also influence tumor development and tumor immunity. After binding and cooperative interaction with T cells, your B cells will actually undergo transformation into the plasma cells. And of course, your plasma cell function is to synthesize and excrete those immunoglobulins. There is also something known as a K-type lymphocyte, and these are mononuclear cells that will actually kill target cells synthesized with antibodies that have been engaged by their Fc receptor. Now, obviously, as we've discussed many of these different alterations that take place, you can kind of start to see and understand why we see such a variety of presentation or morphology when we look at these lymphocytes microscopically. So you can see, you probably have seen already through your hematology class, where a lot of these lymphocytes will take on some weird looking shapes that are a little bit outside of what we normally think of when we think about a lymphocyte with that small size. But you can see now how that process with the immune system can cause those lymphocytes to take on some weird looking shapes. Immunological disorders can actually be divided into primary immunodeficiency orders and acquired or secondary disorders. And then there's also a third category of diseases that are mediated through immune mechanisms that are also included. So because of their complexity and contemporary importance, primary and secondary immunodeficiencies will actually be discussed a little later.